Hello, my name is Anne Howell from the University of North Florida. In this work, we study whether machine learning techniques can recover meaningful metabolite response patterns from a small multimodal dietary intervention dataset, and whether pre-trained microbiome representations can improve that prediction, even when direct sequence information is unavailable. This project starts from a simple biological idea. Gut microbes can alter the chemical environment of the intestine, and those changes may affect human health through larger community level patterns. Metabolites are central here because they give us a measurable signal of that process. Those challenges matter in the context of the original study. The data come from an 8-week fermented vegetable intervention with samples collected before and after the study. The dataset included ASV abundance data, metabolites, nutrients, biomarkers, and demographics. Even though it was biologically rich, conventional analysis did not recover a strong interpretable microbiome signal. This led to the idea of using a pre-trained microbiome model. A model trained on much larger microbiome datasets will have already learned broad patterns, so a small study like ours can build on that foundation rather than starting from scratch. But there is a major obstacle. The original DNA sequence files were unavailable, so we could not use the model in the direct sequence level way it was designed for. The project then became centered on two questions. First, can machine learning recover biologically meaningful metabolite response patterns from this dataset at all? And second, if the original ASV sequences were missing, could we design an approximate taxonomy to model mapping strategy that would still allow us to leverage a pre-trained microbiome language model for meaningful metabolite prediction? For our baseline comparison, we trained a multi-output XGBoost regression model using ASV abundances, nutrients, biomarkers, and demographics as predictors, with metabolite concentrations as the regression targets. We compared this with a simple 1R baseline. The result was that select bile acid outcomes were observed to have recoverable signal at a statistically supported level. Moving then towards the pre-trained model related research question. Because the original DNA sequence data were missing, we needed a systematic way to link the study bacteria to the pre-trained model. We used what we are calling hierarchical taxonomic backoff. Because the biological classification is organized in nested levels, unavailable sequence level matches were replaced by broader classification levels. In this study, that meant genus level matches when possible, family level matches otherwise, and exclusion of features that could not be matched reliably. This gave us a controlled but approximate way to construct pre-trained microbiome features. The result of this strategy was that most of the dominant abundance signal was preserved. We then passed the HTV-derived sequence arrays through a pre-trained Electra-based microbiome encoder. A supervised regression head was trained on top of this representation to predict bile acid outcomes, and its performance was compared against the same 1R regression baseline. The result was that the HTV Electra model also showed statistically supported improvement over not only 1R for the selected bile acid tasks, but also the baseline multimodal model. The surprising result from research question 2 was that HTV Electra was not only usable as an input representation, but that it could function as a standalone regression model. Unlike the multimodal XGBoost baseline, this model did not use the broader study feature set, such as nutrients, biomarkers, demographics. Even with that narrower input space, HTV Electra produced statistically supported improvement over 1R for selected bile acid tasks. Our strong results with respect to research question 2 brought about a new question. If XGBoost was learning from raw ASV abundances plus the broader multimodal feature set, while HTV Electra was learning from a transfer pre-trained microbiome representation alone, were these models capturing any complementary structure? To test this, we used a fixed 50-50 prediction level stack. The result was that the stack model produced more statistically supported improvements for the selected bile acid tasks relative to the multimodal baseline compared to the HTV Electra model. The implication then is that HTV did more than make the legacy data compatible with the pre-trained model. It also offered a complementary form of computable microbiome knowledge. Thank you for your time. Are there any questions?